My dear students, let's complete 8.5 translation process. Again, go back to the reading frame. This reading frame, where can I find this reading frame? This frame on the mRNA exactly. How many codons are there in this reading frame? What should I do? I should count first. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 divided by 3, 4 codons exactly. Those are 4 codons for 4 amino acids. ARG, TYR, SER, SER. Those are the abbreviations for different amino acids. This is the reading frame 1. So the question now, what will happen to the frame codon if there is change in the order of nucleotides? What might happen? They found that a change in the order in which codons are read changes the resulting protein. Of course, try to imagine that there is a change in the codon. There is shift in the codons. So instead of starting from CGA, starting from GAU. So look how the amino acids are completely different right now after only shifting of the nucleotides. So any change in the sequence of the nucleotides might affect only one codon or more, or might affect all the frame. And this is one type of mutation. Might affect all the frame, and these are mutations. So the question now, why are genetic codes universal? What's the meaning of universal? So you can apply the same sequence of codons in all organisms. How? Look here. CGA for arginine, is it in human or in ants, yeast? In all organisms. So CGA, this codon for arginine in all organisms. That's why it's a universal codon. It can be applied in all organisms. AUG, it's the start codon in only one organism? No, in all organisms. That's why we can say that those genetic codes are universal. So regardless of the organisms, codon code for the same amino acid in all organisms. Now, if I consider the process of, or the formation of protein happens in a factory, what are the main machines that should be in this factory? Let's say that the cell has a factory for forming proteins. What are the machines inside the cell that can form the protein? First of all, where will this factory be? This factory, yes, of course, in the cytoplasm, because as you know, the translation process takes place in the cytoplasm. What are the different appliances, or what are the different machines in this factory? Number one, as I told you, mRNA. This is mRNA, which carries the reading frame for amino acids. Number two, what is this? Do you remember? Yes, exactly. It's tRNA. tRNA is the second machine. The third machine is the ribosome. So ribosome, tRNA, and the reading frame or mRNA, those are the machines for this factory, which is the factory for forming protein inside the cell. So now you know what the function or what the role of mRNA. It carries codons that will be translated. Okay, which other machines will translate or will read those codons? Number one, tRNA. What the function of tRNA? If I asked you what carries free floating amino acids from the cytoplasm to the ribosome, I told you tRNA, it's like a truck. It carries amino acid. It carries amino acid. Which amino acid? Any tRNA can carry any amino acid? No. The tRNA with anticodon that is complementary to the codon for this amino acid. Did you understand? Let's know. Now here is a tRNA, and this is mRNA. It carries codons. CAC, this is a codon. Which tRNA of those can bind to this codon? CAC. The tRNA which carries the 3 nucleotides that are complementary to those nucleotides, which are called anticodons. Those nucleotides which will form anticodon. So GUG is an anticodon for CAC. The C pairs with G. A pairs with U. C pairs with G. I didn't say A pairs with T because it's tRNA and this one is mRNA. So I'll not talk about T at all during talking about mRNA. So CAC is a codon. GUG is the anticodon. The anticodon is attached to tRNA, which carries the amino acid for that is coded by this codon. UAU, which anticodon combined to it? U pairs with A, U, A. So this tRNA carries an amino acid that is coded by UAU. You can look at the table and know which amino acid can be carried by this tRNA. UAU. AUG. So of course you know now which amino acid is carried by this tRNA. Methionine. This is the start codon. AUG, methionine, for the methionine, which is the start codon. And this tRNA has anticodon UAC. So now you know the role of tRNA. It's like a truck that carries amino acid, and it has an anticodon. The anticodon binds to the codon that is complementary to the nucleotides in the anticodon. The third machine is the ribosome. This is the ribosome. Let's talk about the structure of the ribosome. Small subunit. This small subunit binds to mRNA. This is the small subunit. And large subunit. The large subunit, it has 3 binding sites. Those 3 binding sites to bind to what? To bind to tRNA after carrying the amino acids according to the codons on the mRNA. So there are 3 binding sites. This is the exit site, and this is the second and first site. This is the ribosome, which is the third machine in the factory of protein synthesis by translation. For translation to begin, the tRNA binds to which codon? Start codon. So if I told you the start codon is AUG, so the tRNA will have anticodon. I told you AUG, so UAC exactly. So a complementary tRNA molecule binds to the exposed codon, bringing its amino acid close to the first amino acid. Let's see together. This is the first step. Now, methionine, yes, this is the start codon, and this is the tRNA that carries methionine, and it has anticodon UAC. The small subunit to bind to the mRNA. Large subunit, it has 3 binding sites to bind to the tRNA. The first tRNA will bind here in the second site carrying methionine. Now, the first site will attract the tRNA, which carries the anticodon complementary to the second codon, which is, for example, leucine. So now, leucine, the tRNA that carries leucine, will bind to this codon. After that, the ribosome will start to break the bond between the amino acid and the tRNA, and will form a bond between the two amino acids together, which is the peptide bond between the two amino acids. And this is another role for the ribosome. After that, the ribosome will start to pull the mRNA strand to let this site free, empty, to attract another codon. So it will pull the reading frame. So after that, the first tRNA will go to the exit site and will leave the ribosome. After linking the two amino acids together, now this codon is exposed and will attract another tRNA that carries amino acid, because of this tRNA has an anticodon complementary to the codon, and so on. After that, this tRNA will go to the exit site and again shift and again pulling of the mRNA till what? Till finding a stop codon. This stop codon means stop translation. No more amino acids are needed for this protein.